Hello and welcome back to another exciting episode of Ed Puzzle Notes as we continue our journey through the world of meiosis. Later on, we will get into genetics, which I don't know, maybe you'll like. Anyways, so meiosis, Meiosis, it also has that phase called interphase. So this is when these cells in your body are not replicating. This is when they're just being a cell. Now, in meiosis, we're talking about germ cells or germline cells. So these are cells in the males, they're found in the testes, and the females in the ovaries. And these germ cells, when they divide, they're going to become the gametes, sperm and egg. So germ cells are what's going to, when they divide, they're going to become the gametes. Now, just like in mitosis, so we have homologous chromosomes. Hey, there's a red chromosome. That must be from mom. That's why I made it red. And here's its homologous pair, the blue one from dad. Notice they're the same size. We'll say this is chromosome pair number 15. So they're the same size. They also carry the same type of genetic information. So if this was 15, for example, both of these chromosomes, the blue and the red, would have the gene to determine if you have blue eyes or not. The one from mom and the one from dad. They both have that gene if this is chromosome number 15. They would also have a gene for what color hair you have. Both of them. The one from mom, the one from dad. They both have that gene. Now, in interphase, we remember both of these chromosomes need to make an exact copy of themselves. So that blue one from dad, it makes an exact copy of itself. They're now sister chromatids, and they're attached at the centromere. And the one from mom also has to make an exact copy of itself. So now we still have two chromosomes there, but remember we have four sister chromatids. So we have 1, 2, 3, 4 sister chromatids. And remember, we're going to make 1, 2, 3, 4 gametes or sex cells. Now each one of these sister chromatids, each little arm on these chromosomes is going to wind up in one of these 4 sperm cells or egg cells, the gametes. And it's totally random which one they go in. So this arm right here, the blue one, I don't know, we'll say it ends up in that gamete. This blue arm, we'll say it ends up in that one. It's totally random. You don't know which one they're going to go into. We'll say this red arm comes over here and ends up in that sperm or egg cell. And this red arm on the far left, we'll say it goes into that one. So each one of these 4 legs, those 4 sister chromatids, are going to end up in one of the 4 sex cells, the gametes. All right, prophase 1, the first prophase in meiosis. We're going to have a prophase 2 here a little bit later. But, so this is the same thing, basically the same thing as in mitosis. The nuclear membrane disappears and the chromosomes become visible in a microscope. So you can see the chromosomes right there. Remember these red structures? Those are the centrioles. They're going to pull the chromosomes apart during anaphase. But what I really want you to notice are these chromosomes right there. The red and the yellow and the green and the purple. You can see that they're kind of, I don't know, their legs are kind of crossed. Well, that is a process called, and this is going to be extremely, extremely important, crossing over. This is where these homologous chromosome pairs, so for example, we'll say the red one there is from mom, and the yellow chromosome is from dad. Well, that red chromosome right there is going to give some of its genetic information to the chromosome from dad. Same one as the one over here. We'll say the blue one is from dad and the green one is from mom. Well, these 2 chromosomes here, through a process called crossing over, they're actually trading genetic information. So some DNA from the blue is going to the DNA from the green, and with the yellow and red, some yellow DNA from dad is going to get stuck onto the red chromosome from mom. What is the significance of this? It is a big source of genetic variation or variety in organisms. Basically, crossing over is one of the reasons why, if you have 6 kids, they are not going to look exactly the same. You know, unless you have identical twins, your kids will all look somewhat different. They'll have different traits. One of those reasons is because crossing over increases genetic diversity. All right, so a little diagram that kind of shows you crossing over again. We'll say that the clear chromosome is from mom and the one that's kind of faded in, that's from dad. Well, first, of course, the chromosomes make an exact copy of them, so we have 2 from mom now, 2 chromatids from mom, 2 from dad. If there was no crossing over, and we're just going to make 4 gametes, you can see one of these gametes, these sex cells, is going to get, a full chromosome from mom. The other one's going to get a full chromosome from mom. The other 2 gametes are going to get full chromosomes from dad. Well, with crossing overs, you can see it's happening right here. The mom and the dad chromosomes are exchanging genetic information right here. Their little legs of their sister chromatids are crossing over, and you can see it here. This dad chromosome got a little bit of mom DNA, and this mom chromosome that you got from mom got a little dad DNA. So now we got 4 totally unique chromosomes. And now when these guys separate and go into their individual gametes, now each individual gamete is going to be totally unique. All right, the next phase, of course, we still have our PMAT, prophase, metaphase, anaphase, telophase. Well, this is Metaphase I, the first of the metaphases. And the way you can tell it's Metaphase I is you look, there are 2 lines of chromosomes, basically 2 lines of X's. If this was Metaphase II, it would look something like this, where we'd have maybe 1X mom chromosomes, 1X dad chromosomes, and that's it. So you can see the difference. 2 lines of X's here, only a single line of X's here. Single file line of X's versus double file line of X's. Now we're going to talk about this thing that's super important as well. It's called independent assortment. And let's see if I can explain that. So, during Metaphase I, all of the chromosomes line up in the middle, just like metaphase during mitosis, but it is totally random how the chromosomes line up. For example, on this top cell up here, that top one, you can see that they're arranged big blue, big red, and then below that, little blue, little red. So, if those guys, when they separate during anaphase, the 2 red guys are going to end up over here, and the 2 blue guys are going to end up over here. Now, that's one way it could have happened. On the very next one, you can see it happened a little different on this next cell right there. On this one, we got a big red and a little blue. And on the other side, we got a big blue and a little red. On this one, it's different again. You can see this big blue guy is going to come that way, so there's going to be a big blue in here. And this little red guy is going to go that way. So we're going to have a little red in there. On this one, the red goes that way, so we're going to have a big red. And the little blue guy goes that way, so we're going to have a little blue. So the whole point of this story is all these gametes are going to be totally unique because you don't know how these chromosomes are going to line up in the middle. It's not going to be all blue chromosomes on the left and all red on the right. It's going to be totally random how they line up. Therefore, it's going to be totally random which gametes these chromosomes end up in. All right, the next phase in the PMAT is the A in PMAT for anaphase. And this is anaphase 1. Not going to say a whole lot here. The main thing you need to know, the difference between anaphase 1 and anaphase 2 is that the sister chromatids remain together. For example, if this was anaphase 2, it would look more like this. So the red guys would be separated from each other. Same with the yellow guys would be separated from each other. And the spindle fibers pulling them. So you can see that these are single-stranded chromosomes. They've been ripped apart from each other. Where in anaphase 1, the sister chromatids are still together. So you still have that like X shape. And on this slide, I want you to remember the color blue. I want you to remember the color blue. Write it down if you have to. Blue, blue, blue. All right, telophase 1, not going to lie to you, not one of the more important ones. Telophase, the end of PMAT. Just know at the end of telophase and at the end of cytokinesis after telophase, we're going to have 2 cells. We're going to have 2 new cells. Now remember, in meiosis, we're not making 2. We got to go through meiosis one more time so we get 4 cells. All right, and the good thing about prophase 2 is it's not as important as prophase 1. There's not a whole lot to remember. Just remember that the chromosomes become visible again, the nuclear membrane dissolves, and it's basically exactly the same as prophase in mitosis. But one thing I want to add to this slide, we remembered the color blue. I want you to remember the number 4. 4, 4, 4. Write it down if you have to. All right, metaphase 2, basically the same as metaphase in mitosis. You can see that the X's or the chromosomes are now in a single file line down the middle of the cell. When we think of metaphase, we always think of middle because they're lined up in the middle of the cell in a line. Now remember, if this was metaphase 1, it would look more like this. You would have 2 reds and its homologous pair, the blue guys, line up right next to each other. And then maybe we'll go a green and its homologous pair of chromosomes, yellows, lined up next to each other. And then the spindle fibers would be like this. But, so metaphase 1, you have 2 lines of chromosomes lined up in the middle. Metaphase 2, you have 1 line of chromosomes lined up in the middle. And I know you can't believe it, but we're almost done. We're up to PMAT number 2, the A in PMAT number 2, anaphase. So you can see this is, when we think of anaphase, we always think that the chromosomes are moving away from each other because they both start with an A. They're moving away from each other. Now remember, the way you can tell the difference between anaphase 1 and anaphase 2 is that here in anaphase 2, the sister chromatids have been ripped apart from each other. So if this was anaphase 1, it would look more like this. So you'd have 2 red chromosomes still stuck together and maybe 2 blue chromosomes still stuck together, and they're getting pulled away from each other. But you can see there's still 2 sister chromatids. That's how you know it's anaphase 1. Anaphase 2, it's no longer 2 of them stuck together. It's a single strand chromosomes being pulled apart. All right, so we have made it to pretty much the end. Telophase 2, basically the same thing as telophase in mitosis. All the chromosomes have arrived at opposite ends of their 1, 2, 3, 4 different cells. And with the final process of cytokinesis, remember that's when the cell membrane or the cytoplasm pinches off, we will get our 4 totally unique cells. Remember, we call these haploid. because they have half the original number of chromosomes that we had to start with. So basically, in humans, we start with a cell with 46 chromosomes, and at the end of meiosis, we're going to get 4 cells, each with 23 chromosomes. In case we didn't know, 23 is half of 46, and that's what's called being haploid. Part. Fuck me. All right, so 2 little diagrams that kind of show the difference between mitosis and meiosis. Up top, up here, we got mitosis. So we start with a very simple cell, 2 chromosomes, 1 blue from the male and a red chromosome from the female. And at the end of mitosis, we get 2 cells, which are exactly the same as the original parent cell. In meiosis, it's a little different. We got 4 chromosomes in here. 2 reds, a big red from mom, a little red from mom, big blue from the father, little blue from the father. And by the time we get done with all the phases of meiosis, you can see we've cut that number of chromosomes from 4 to 2. So we've cut it in half. And you can see that each one of these cells, these gametes or sex cells, are totally unique. Now, I hope that you can see that some of these chromosomes have a little bit of blue and a little bit of red. Well, remember, that was from the important process that occurs during prophase 1, crossing over. So now, these gametes are even more different than they would have been because some of these genes in the DNA have been traded from the female chromosomes to the male chromosomes.